I've been thinking about old-growth mycorrhizal communities for a long time, but it's only very recently that I've had the opportunity and the ability to start studying them. It's much harder than I thought it would be. First of all, it's really hard to find these communities. You'd think it would be easy, but it's not. And second of all, it's really hard to interpret what's going on. But before I start, I'm going to show you some experiments I'm working on right now. They haven't been published. They're early days. But before I show you this research, I think we have to start earlier. We have to go back, quite a ways back, to really understand the story I'm telling you today. Okay, I don't know what the resolution is going to be like for some of these photos. They're very old. I'm not sure how many of you have been to Vancouver, British Columbia. It's a big city. It's about 2 million people. I love this photograph because this is pretty much downtown Vancouver, early 19th century. I love this photo, and I love all old photos. I look for old photos all the time because they allow me to get a sense of what it was like before humans really stomped all over the planet. Because we don't know, right? We have this sense we know what has been lost. But I think we grossly, grossly underestimate how much the world has changed. I do this all the time. No matter where I am, I look out the window or I'm in a new country and I just imagine what it must have been like before humans were here. On the train from Paris yesterday, looking out the window in the beautiful countryside of France, the fields, the villages, but can you imagine what it was like to be here? What it felt like when there was a continuous forest from Sweden down to the Mediterranean? Oh, we have to imagine, right? Because Europe lost its forests so many thousands of years ago that nobody really documented what they were like. And there were certainly no photographs around at that time. Even where I'm from, where the impact has been very, very recent and very quick, we still have such a poor idea of what's been lost. I mean, there are a few photographs, but like I said, I think we've lost more than we can possibly realize. Okay, so this is a picture of Vancouver, early 19th century. This is around Granville, if you know Vancouver. Very close to the spot, but about, wait for it, about 50 years later, same spot, a little bit later, still 19th century. See some houses, trees are gone. And this is Granville today. In 200 years, biodiversity just annihilated. It was completely gone. And what's been lost? You know, it's obvious, right? The trees have been lost. Those big, giant trees, they're gone. How these guys did it, I don't get it, right? These men, from California to Alaska, they removed the forests in like 100 years with just their hands. They didn't have power tools. They just cut them down. So it's obvious what's been lost. The trees have been lost, but they took them all. And they didn't just take the trees. They took the plants that grew with the trees. They took the lichens. They took the moss. They took the birds. They took all the animals that grew in these forests. But they took something else that all of us here know about, but no one talks about. We all know that the forests are above ground and below ground. What happened to the forest underground? Is it still there, or has that been lost too? This is what I'm going to talk to you about today. Okay, I study arbuscular mycorrhizal fungi. Most of you probably know what they are, but it's a pretty broad group. This is a picture I took of a plant root. The big white thing is a plant root. The tiny white things are the hyphae that are coming out of the root. Arbuscular mycorrhizal fungi are fungi that are obligate biotrophs. They have to live inside of plant roots. Here's a plant cortical cell, and you can see, I hope, an arbuscule. That's the hyphal structure where nutrient exchange and carbon exchange takes place, right? The fungi collect nutrients for the plant and exchange it for host carbon. Now, I'm going to talk about arbuscular mycorrhizal fungi in these old systems. But I want to tell you a little bit about this group of fungi, the Glomermycota, that's their phylum. They're really species poor. I mean, compared to the plants they grow with, plants have what, 400,000 species minimum? Glomermycota have 250 described taxa, and that's low even when you compare it to other closely related groups, like the ascomycetes have what, I think 60,000 described taxa. They're species poor. Even though we're accumulating new data, and there's more DNA species every day, still, they're not very species rich. And people have said this is because, well, they're generalists, right? They'll associate with any plant that's out there. They're ubiquitous. You hear that all the time. They've got a global distribution. But do they? Do we really have the data that we need to make these kind of statements, these tenets of arbuscular mycorrhizal biology? I don't think so. I think maybe our knowledge has been seriously biased by the way we've been collecting data. There's pretty much two ways you can look at mycorrhizal fungal communities and mycorrhizas in general. You can trap them with trapped plants. So you go out, collect some soils, and then grow it in a greenhouse with a host plant, usually a cultivar, a plant like that. But and that's a great way of getting material to work with. You can look at the fungi, you can describe them, you can see how they grow. But that method, these fungi are desperate, right? You've got one plant for them to grow with. So you are, by definition, culturing only generalist fungi, a fraction of the community that's out there. Okay, so that's one huge filter on the community. If you go out into the field and you sample from old field communities, that's great, right? You're seeing what fungi are like in nature, and you can sequence their DNA, and you can do all sorts of things with it. But typically, up until very recently, almost all our data from natural systems came from agricultural or formerly agricultural sites. And these fungi growing in these systems, these former agricultural sites, these are the fungi that don't really mind having their mycelium plowed out every now and then. They don't really mind growing with annual crops. Again, a huge filter on potential biodiversity. So I think, yes, maybe our understanding of this phylum has been constrained by the way we've looked at it. And maybe we just haven't looked broadly or deeply enough. And this is a basis of a study I did this year with some collaborators. And we asked a question. Wait, it's coming. The question is coming. Are there more undiscovered AMF in natural ecosystems? And the thought behind this was, because we've looked so deeply at disturbed systems, agricultural systems, maybe there's so few taxa because we haven't looked at very many different types of systems. So we performed a meta-analysis. We combed through all the molecular studies we could find and categorized them by disturbance regime, whether or not they'd been impacted by human, like agriculture, mining, or if they were a natural ecosystem. And overwhelmingly we found, yes, natural systems have more undiscovered fungi in them compared to human-impacted ecosystems. Now, whether or not, who knows exactly why this is happening? You know, natural systems have different kinds of plant hosts. They have different disturbance regimes, different nutrient profiles. There's all sorts of reasons why this could be happening. This is really slow. So things like forests, grasslands, desert, shrublands, these systems, they're primarily made up of fungi that have not yet been described yet. So yeah, we've been looking at too few hosts in too few ecosystems. So maybe old growth AM fungi are out there. Maybe they exist. We certainly don't know about them yet the way we've been looking at Glomermycota. So I decided the best way to look at this would be to look at this tree, Thuya plicata. This is western red cedar. It's one of my favorite trees, but it's an excellent system for looking at mycorrhizal succession, in my opinion, for the following reasons. Number one, in British Columbia at least, we have a really good idea of how long cedar has been there, and there's a good chronosequences of, since the glaciers retreated from BC, cedar started appearing in different pockets. So we know that they have been in BC from about 100,000 years ago to the most recent stands are about 500 years old. So that's a really good long chronosequence. Number two, AM hosts long-lived AM hosts are rare in temperate ecosystems. In tropical systems, not so much, but in Canada, Thuja plicata is pretty much the only long-lived tree species that hosts just AM fungi. And like I just said, they're very long-lived. They can live up to 3,000 years old. So for me, they're the ideal model for looking at succession in mycorrhizas, but they haven't really been studied. I think there's one study on Thuja and one on Sequoia. They've really been understudied. So the next, right now I've got 2 studies that haven't been published, 3 studies. I'm going to show you data from 2 of them about my work on Thuja. The first one is we decided to sample with Monica Gorzlak and Dr. Brian Pickles. We looked at Thuja from its northernmost range and its southernmost range, and we asked the question whether or not tree age mattered. Would we find earlier successional AMF communities in young trees versus those really old giants? Now, it's impossible to accurately age these trees because as they grow, they start rotting from the inside, so you can't core them. So our proxy for age was diameter. And so the big old trees versus the intermediate age versus the juvenile trees. So we extracted DNA from the soil and the roots around these trees, and we analyzed it, and we found no real consistent difference among age classes. And I was surprised because I'd looked at other plants and found age differences over the lifespan of the plants, and other people have found this too. I didn't find this convincingly for cedar. I mean, there's an indication that there are specific fungi only in the old trees, but rarefaction, the way I rarefied my data, had a huge effect on whether or not this was always significant or not. So there wasn't a strong trend for tree age. And like I said, I was surprised, but I thought, well, the truth is these stands are very, very old. Each of these stands, the northern and the south, are about 3,000 to 4,000 years old. And we know this from pollen data. So it's possible that these communities were already climax, right? So the species pool was set. There's not going to be a lot of variation in the, within ages of trees. So I decided maybe it's not tree age. Maybe it's something else. Maybe it's exposure to disturbance. So the next experiment we decided to do is we stayed just in the southern range, and we looked at trees that had been recently disturbed, or stands. So it's no longer about the trees, it's about the age of the stand. Does the age since disturbance matter? So we looked at clear-cut versus old-growth stands. And this is a project I'm working on with 2 students, Dylan Zaitzoff and Mika Vanderheid. And what we did is we paired recently clear-cut sites, less than 5 years, to old-growth sites. And we don't really know how old those sites are, but they're primary forests. The stands have been there for thousands of years. Okay, just keep going. And again, we sampled, here we just sampled the soil because obviously there's no cedars left in the clear-cut. They're all gone. So what did we find in this case? Well, in this case, just the soil community, there is a huge difference among the clear-cut and the old-growth sites. And okay, so this is just an insight into what was changing. These are all taxa, AMF taxa, that were significantly different among the old-growth and the clear-cut sites. So above the line, they were dominant in old-growth, and below the line, they were dominant in clear-cut. And you can see there's a mix of glomuses, big surprise, but there's a lot of unknowns. But what does this really tell us about the old growth communities? And this is where I feel like I'm hitting a wall with mycorrhizal research. Just collecting these species inventories isn't giving us enough information about what these communities mean, what they do. I think everyone probably in the audience who studies soil organisms is in the same boat. We don't have enough information about the biogeography of these organisms to be able to properly interpret this. I would love to look at that and say, oh, that's an indicator of an old growth site. And maybe someday I'll be able to do that. I'm starting to feel like I'm just not going to live long enough to be able to see this research project through, because I can't even get close to making those kind of proclamations about these communities. And that's what I want to do. I want to identify, I want to have a picture of a spore saying this is an old growth indicator species. That would be such a powerful tool to be able to go into ecosystems and have that kind of insight. We're very, very far away from that yet. Now, I don't want to sound too pessimistic, because I know of so many groups around the world that are working hard on this and are accumulating information, but we still can't properly compare our results. We don't have enough information yet. So where does that leave us? I think based on these preliminary results, there's something going on, right? Old stands versus young stands, old trees versus young trees. The communities are different, they're changing, but it doesn't mean we know anything about old growth fungi. We're starting to know something about old growth forests, but we don't know anything about old growth fungi. What we really need to do is find out, are these fungi ecologically different? Are these fungi functionally different? Now, do they grow differently? Are they stress tolerant? Are they more competitive? Are they slow growing? Do they tolerate really low levels of host carbon? What kind of symbionts are they? Are they cooperative or not? I guess it's working again. I want to answer those questions, but to do that, I have to be able to culture these fungi. So I've started these little cedar seedlings in my lab. Do I have to wait 2,000 years to cultivate the species that grow on them? I hope not. I hope not. I guess all we can do is see. The key now is to really identify where we're going to find these old growth fungal communities. I think I have to be really careful about how I choose them. It's not old trees. I don't know if it's old sands. I think it's old soils. But talk to a soil scientist, talk to 5 soil scientists, and they'll give you 5 different definitions of what an old soil means and where you might find it. So that's my task right now. I think I've got a good chronosequence that I'm going to sample this summer, but I'm not sure. And the last thing I want to tell you, pedicide. I chose that word deliberately because I think that's what's going on. We've lost the trees, they're gone. Definitely, within our generation, all of these old trees are gone. What about the rhizosphere? Have we lost that? Even if there are refugia that are left, have they been so inundated by the surrounding communities, the surrounding disturbed communities, that we don't even have them anymore? And if we decide in the future that these old communities are valuable and we want to resurrect them, have we lost the ability to do that completely? And that's all I have to say today. Thank you.
Apologies to Miranda about the vagaries of the timing device. So can I have one question? Can we bring the microphone over here? Just stand up and talk as loud as you can, I guess. Sorry, I don't know when the microphones are. Yeah, go ahead. That's right, there's one coming.
Hi, thank you for this brilliant presentation. I like the passion to which you describe this like pedicide and this killing of thousands of trees. My question is not very much related on your research in particular, but more on the way that forest trees are exploited in Canada and in other parts of the world. Don't you think that there are alternatives to the clear cut in the forest exploitation? Because I think in other parts of the world we are already adopting different systems that
Definitely. I mean, there are schools of forestry around the world and in Canada that have shown that there are better alternatives. I don't know what's wrong with Canada. I'm constantly ashamed of what's going on. The 10% of old growth that's left, most of that has been licensed to forest companies. I can't even believe it's true, but it's true. There are old growth forests in Canada that have been flagged for logging roads and harvest, and you're right, it's clear cut. I don't know. I wish I had the answer. It's abysmal. It's a shaming.
Thank you, Miranda.
All right, thank you very much.